unexplained weight loss. So these are some of the major symptoms associated with esophageal cancer. And now let us look into the stages of which the cancer is being distinguished. So there are 4 stages: 0, 1, 2, and 3 and 4. So it is based on TNM model. So T stands for tumor, the growth of tumor. N is its extension to the lymph node, and M is to the metastasis, that is to the distant organs. So here we can see at stage 0, the cancer is in the epithelial lining in T1, right here, and stage 1 and 2, where it is growing out of the epithelial cells and it is going to the esophagus. And next, it will spread to the nearby lymph nodes and finally to the other distant organs. So these are the different stages of esophageal cancer. And now, after understanding the basic information, now let us see what are the current challenges we face. So first challenge is the esophageal cancer is asymptomatic. That is, the major symptoms associated with this cancer is not seen until the cancer is progressed by 70% and till it is in the later stages. And hence, we need early diagnosis and early treatment. And for this early diagnostic method, there's also already early method which is being invented. It is called as cytosponge, and this is done very recently, like in 2018. It is a minimal invasive method. So there is already a method which is being established, and now we need to have a treatment procedures, and there is a lot of research that needs to be done to accumulate the data on the early stages of cancer to understand what is happening at the molecular level to know the cancer progression. And hence, we are using a multi-omics data approach for understanding the cancer progressions. And now, we will discuss about the oncoproteogenomic approach to understand the cancer mechanism. So, proteogenomic is a technique of analysis where we are combining multi-omics such as genomics, transcriptomics, and proteomic data to improve the gene annotations. So here we are putting, aligning DNA, RNA, and protein in central dogma perspective. So we'll be linking all this information to understand how the prognosis of cancer is happening. And another important aspect of proteogenomics is it also captures the PTM information along with the variations here. Even the PTM information can be tagged along to know the cancer prognosis and hence the pathways that is involved in the process. This is the application part of proteogenomics where it is applied in precision and personalized medicine. Now, the PPM is the future of cancer treatment. It has already been done on different types of cancer where the precision or personalized medicine is being, the approach is being used for cancer treatment. And these are the 3 methods of precision medicines which can be used for cancer treatment. But we don't have sufficient data for esophageal cancer, and hence we can use from our research, we can try to extract this data and which can be in future can be used for precision medicine. So based on the current requirements and challenges which we have seen, now we are framing our research objectives into 4 sections. So first research objective is to identify and annotate the novel genes and peptide sequences in esophageal cancer. Second is to identify the novel initiation codons, splice isoforms, exon extensions, and SNPs. Third is to identify the novel post-translation modifications associated. And fourth is to generate a proteogenomic map for esophageal cancer samples. Coming to the first objective, that is to identify and annotate the novel genes and peptide sequences in esophageal cancer. So this is a background of what is a novel peptide. So novel peptide is something that sequence of the peptide which is not present in the reference protein database. That means the reference protein database already has a curated set of peptides. So we are looking at the missed information at the genomic section where it can be expressed at the proteomic level. So we are looking at some information which we can tag along to the genomic section. So this is called as the novel peptides. And how are we going to achieve this? Is by using two omics data. So high throughput genomic whole genome data and high throughput mass spectrometry data is being used. And we are going to generate a customized database for each of the searches, and then we are going to identify peptides and its associated gene, which can be further used for targeted gene therapy. So this is the workflow which we have used to obtain the novel peptide sequences. So this is divided into two sections. This is the first section and this is the second section. So second section is basically a proteogenomic mapping, and first section is to eliminate the already custom protein sequence data. So the mass spectrometry data was obtained for initial stages of cancer from PRIDE archive, and it was converted using MS Convert into the readable files, MGF files, and then it was mapped with the custom protein sequence already available database. Some 20,000 odd entries was matched, and whatever unmatched spectra was further used for proteogenomic analysis. And in the second section, we are using a SRA, high throughput whole genome sequence data. So this we took around 10 whole genome sequence data from SRA database. The quality check was done, and this was used to create a custom database. So what are we doing here is the whole genome sequence we are translating into the possible frames. So every sequence of the nucleotide can be translated into 6 possible, 3 forward and 3 reverse frames. So we translated the entire whole genome into 6 possible frames using emboss tool, and then later into the peptide sequences, so we could do the proteogenomic mapping. So we accumulated around for 10 of the whole genome sequences. Once it was translated, we got a huge protein database which was almost around 1 TB. 1 TB of data was translated, and not to miss out of any possibility of possible translations, we did this translation and we recorded it into a database. And next it was used for proteogenomic mapping using expanding tool. So once mapped, we got 16 distinct novel peptide sequences which was specific for esophageal cancer. So this was the 16 sequences which we obtained. And once we got this novel sequences, we had to map it to the gene or the genome and where it belonged to. So for this, we used a tool called as ACTG tool, where amino acid sequences are going to be mapped to the genome, and hence it was categorized into 3 sections: long non-coding RNAs, protein coding regions, and pseudogenes. So this is the 16 peptide sequences which we obtained, and it belonged to the 2 genes. So one is the LEC1 gene and the location, and second one is C16 or R62 gene. So these are the peptide sequences which we obtained after searching. So in detail, the first gene is associated with methylation, and it is associated with the cancer progression, and it also has functions in mutation, deletion, and inactivation of proteins. So the further investigation of variations or the PTMs will be done in the next objective, but right now we have only identified the novel peptide sequences for this gene. And this is the second gene, and where it codes for BPS35 protein, and it acts as an intracellular protein, and it functions as a membrane protein as well. And this particular gene has a prominent role in activation of transcription factors, 12 transcription factors. And we went further to know the functional predictions of this particular peptide sequences to know what function it has. So we used one prediction tool called as ELM prediction tool. So based on the MOTIF data, we predicted that phosphorylation was predominantly seen as the function of this identified novel peptide sequences. So coming to the conclusion, by applying proteogenomic approach, we have identified 16 novel distinct peptide sequences for ESCC, and these novel sequences encode for two genes, these two genes, the LDC1 and C6162 gene, and this was not part of ESCC annotations earlier. And we also predicted the functional sites, and phosphorylation was seen predominantly. And here we are recording the minute changes at the genomic level. It's being captured using proteogenomic approach. So coming to the second objective, where identification of splice isoform, SNP and exon extensions. So here, what are the proteomic variations? This is single amino acid variations and novel splice junctions. So alterations in this have a significant impact on the structure and function of protein. And hence it is important to understand these variations. And how are we going to do this is by using the analysis method RNA sequence proteogenomics. And in this section, we are using a transcriptomic data, and we are mining the variant peptides from the transcriptomic data, and we are creating a customized database again. And here we are not including any already curated information from TCGA or CAN or Pro, but instead we are doing analysis from the raw data, and we are mining for the variants and then going for a spectral matching. So this is a workflow which we have followed for mining of the variants and also the spectral matching. So this part is for transcriptome analysis and mining of the variants and also the database generation, and this part is for proteomic study. So both together is being mapped using metamorphics too. The spectral matching is done here. So first, what we did was we extracted raw RNA sequences from SRA database. So 10 different sequences was obtained, and the data was aligned using a Galaxy platform. The alignment was done on using two different tools. One was HiSat2 and TopHat. So this was done to record two different types of variants. So HiSat was done to record SAD peptides, and TopHat alignment was done to record splice junction peptides. So once the alignment is done, what we did was we used a snippet tool and we did a variant calling, and this was again recorded in the VCF files. So this was the first segment in our customized database. The second segment where the alignment is done using TopHat, and we are using two different workflow, that is known junction, known and novel junction, and once we subtract the file, we are only looking at the novel junctions. So this junction is recorded in the BEP files, where the novel junction coordinate is usually translated into peptide sequences using the genomic locations, and this locations is added in the peptide sequence is added in the NSG peptide in BEP files. So this was the second segment, and the third segment is Unicron proteomes in FASTA. So these three elements went into creation of a customized sample-specific database for each of the samples. So 10 such database was created for each of the sample, and this was sent for spectral matching. And here the mass spectrometry data, again the MG files was converted into MGF, and it was sent to metamorphics tool for spectral matching. So these are the results which we acquired after performing the spectral match. So these are some of the single amino acid variants which we have recorded, and we have got around 600 SAD unique peptide sequences, and also in this importantly we have recorded only missense mutations, that is non-synonymous ones, and we have also listed the location, the IDs, what is the change that is happening, and also the count. And coming to the novel splice junctions, here we have around 38 different novel splice types, exon types, which has been identified, and here it has been categorized into protein coding, pseudogenes, and antisense RNA exons. So it is one more important observation here is NE3 is one type which has been predominantly observed. It's malleate enzyme 3. So these were the two important results which we got from spectral matching, or two important proteomic variation which we could capture using the RNA-seq proteogenomics. And coming to the second part, it is prediction of initiation codons. So here we have actually combined three of the prediction tools to know the prediction site or translation start sites. And this helps in the controlling gene expression or the regulation. And the prediction tool used are ATGPR, EST-scan, and NetStat. So the novel peptide sequence which we have identified earlier is used here. And what is the principle that has been employed? ATGPR tool uses triple weight matrix program. The EST-scan tool uses fifth model HMM models, fifth order HMM models. And NetStat tool uses artificial neural networks to identify the star codons. And this is one of the results which we have produced for one sequence, where we can actually see where is the position of the first ATG present, and the prediction done by three tools. But the NetStat is almost predicted the function, but the rest of the tools has predicted properly. So we have done this for all the 16 sequences, and we have combined the location of first translation start set for all the novel peptide sequences. Now let us look into the third objective, that is identification of novel post-translation modifications. So as we know, PTM post-translation modifications plays a very significant role in structure and function of a protein, and hence it is very important to track down the PTMs, especially in cancer cells where there's a lot of changes that keeps happening. And here what are we doing is we are employing a global PTM search strategy, where globally we are looking for all the possible PTMs that is associated. And here again we are creating one specific customized database, and this database we are adding information of site-specific PTMs, and hence going for spectral matching. So this is the workflow which we have followed. And again here, if we see, we have taken RNA sequence raw data, and the alignment was done, and we mined all the variance peptides, and it was recorded in a file. And this file, this particular variance was attached with the site-specific PTM annotated data, and the database was formed. So every variation, whatever was recorded, was also attached with the site-specific PTMs. So this database was further matched with the proteomic data using the Metamorphosis tool, and we could capture all the PTMs that was associated here. So there was totally 161 different types of PTM that were identified using the GPTM search strategy, and predominantly observed was phosphorylation, acetylation, methylation, and hydroxyl. And these were the other types of PTMs that was observed. And in phosphorylation, we had kinase related phosphorylation, which plays a major role, especially in cancer progression. And there was other type, even the tyrosine phosphorylation was also seen, and these were the placement counts. And this was the second type, where acetylation. So this is majorly involved in modification of histone proteins and transcription factor. Hence, this is also important to see how it is expressed in the cancer cells. So these were the two PTMs which were observed, and other PTMs are also being listed out. So finally, after gathering all the information, now finally we are going to produce a proteogenomic map using a tool called as POGO for esophageal cancer. So what is a proteogenomic map? It is used for visualization, first of all integration of all the identifications of novel information, and visualization on the genome browser. And here another important aspect is we can also add PTM information as also being integrated into it, and it also gives us the exact location and that has been modified or mutated to know the gene targets. So this is the working procedure of the tool, where we are given input in format of GTF files, that is the annotations, genome annotations, and the transcript informations, peptide sequences, and what all the other variant information has been given. And once it maps, it is going to distinguish into splicing the name of the gene, the biotype, or the PTM information if available. So this is one of the results which we have done for our sequence. So this is a sequence which we have taken, and it is showing us all the related information, that is what is the peptide that is available, what is the type of medium, where is the position, what is the name of the gene, and the biotype of the gene. Every information will be put in the browser, so it is like a one stop to analyze all the modification of a interested gene target. So this is a view of the proteogenomic map which we have done, and this was the version which we have taken. And here for our selected sequence, we can see here the PTM information also is given along with the sequence, and the biotype or the gene information is here, and also they provide with a SNP changes or the variation changes is also mentioned along with the location. So once we want to know anything about this particular gene interest, if you just go to the browser and upload a map, the proteogenomic map, one browser will give you all the required information for the particular location. The PTM changes or the SNPs or the location and everything or the protein that encodes for it will be available on this map. This was the proteogenomic map which we did. I'm going to conclude the work. So from numerous number of peptide sequences, like as I mentioned, we had around 1 TB of data, which was a customized protein sequence data, and we did a spectral matching and we got only 16 distinct peptide sequences for esophageal cancer, and this belonged to 2 particular genes, and one of the gene can be used as a potential target for esophageal cancer immunotherapy. And the exon tag which we identified, that is ME3 or Manneken-San, can be observed in gene therapies, can be targeted in gene therapy. And the PTMs, that is phosphorylation, kinase-driven phosphorylation, and tyrosine-driven phosphorylation was also seen, along with acetylation was predominantly observed. So, scope for future work. Here, DLEC1 gene, which is already been studied and carried out by our juniors in the lab. They have already been doing some research work on this particular gene. And second one is recently the Asian Genome Reference Consortium has been released. So we are planning to produce a proteogenomic map specifically for population specific for India or the Asian populations. And third thing is to use the new age technologies like artificial intelligence or machine learning to partially automate the workflow and to extend this proteogenomic approach to other cancer types, and also for targeted gene therapy. And finally, to connect different data, that is to connect the imaging data of the cancer with multi-omics data, so that we can analyze the larger cancer data sets. So these are some of the future prospects which we have listed out. These two are the journal publications which we got, and out of this, this one was Q1 publication which we got. And we have 2 conference publications as well. One was international and one was national conference. I've listed out a few references. I'd like to thank Dr. Vidya for giving me this opportunity to for her to accept me as a student and to provide me with all the facilities to complete my PhD successfully. My family, my friends, and my PhD teammates Akshata and Suchitra. We all joined integrated PhD together, and she completed it also. And Sanjay, Akshay, and Vasan, they were our teammates, and he has been a critical reviewer also of my work. And these two are Vidya and Vasika.
I think Vidya and Vartika, they are BTech students.
BTech students. So they helped me with my spectrum generator. And Pradeep sir and Shivkumar from Department of Biotechnology. TechWeb for funding the initial days of a PhD work. And ma'am, because once I went to ma'am's lab for proteomic data, which we were stuck for 3 months, and I took help from her student to solve that issue. And thank you ma'am for that. Now we open for questions.
You have taken whole genome sequencing data.
Yes.
So, how did you translate it? Is it like you assembled it and then made a single sequence and then you translated, or you just translated the raw data?
Yes, ma'am. Just the raw data. So whole genome sequence data, the SRA data is extracted. So SRA data, we know that it has been single right? So as it is translated using a tool called as Emboss. So this Emboss tool gives us all the possible